Can everybody hear me now? All right. Sorry about that. I'm delighted to introduce Michael. If I were Hungarian, I would say something like Szego, but I'm not. He's probably more used to hearing Szego. I'm sorry about my mic. It was working when we chit-chatted, but stopped at the critical moment. So we've asked him to start today covering two genetic topics: gene editing and personal genetic knowledge, as you get when you send your personal materials off to 23andMe. So we gave him a bit more time, but there still will be plenty of time for questions and discussions. He has a superb background for this task. He has advanced degrees both in molecular genetics and in bioethics. And with that knowledge, he's director of the Center for Clinical Ethics, the joint venture of 3 Toronto hospitals, and a research ethics consultant for the University of Toronto Center for Molecular Medicine. Over to you, Michael.
Thank you so much. I'm going to try and share my screen here. Give me one moment here. It's not allowing me to share screen, but it looks like a problem on my end. And I saw this just prior. So Vinise, I sent you an email with the PowerPoint presentation. Are you able to upload it?
Yes, I'll look into that right.
Michael,
Perfect.
Well, I just changed the security precautions. Try again.
Okay. No, it says I have to go into my system preferences. So it looks like a problem on my end, which is strange because it had worked earlier. So Vinise, did you receive a copy of the presentation?
Oh gosh, I'm just looking at it, downloading now. Yes, I did. One moment.
Perfect. Sorry, everybody. We should be very good at this by now. There we go. Just go down to the show screen.
Sorry, one moment. Do you want it to be the slideshow?
Yes, please.
Okay.
Okay, and am I able to? Okay, I'm gonna have to say advance slide.
Okay.
Okay, sorry about that, everybody. So I am here to talk about ethical issues associated with next generation sequencing. So next slide, please. So here's a kind of outline for the next hour. I thought I'd give you a little primer on genomics. The whole lecture could be on this topic, so I'm going to try and keep it very high level. It is actually not that complicated when you break it down, and so hopefully I can make a complicated topic into a relatively simple one. And then we'll talk a little bit about some of sort of the emerging issues that have come out in relation to next generation sequencing and all of the genomic technologies that are available. And those are issues principally related to privacy and confidentiality, what results ought to be returned to either a research participant or a patient. And I'm going to focus particularly on the pediatric context because that's sort of an area where there's been a lot of concern around what kind of information ought to be returned. Talk a little bit about genetic discrimination. We've got a Genetic Non-Discrimination Act here in Canada, and I'll describe a little bit about what that entails. And then finally, I will end off with genomic modification. And so we now have the ability to change the genetic makeup of people. There have been some examples of scientists that have perhaps jumped the gun and done this. So we'll talk a little bit about what genome modification is and what are the ethical issues associated with that. Next slide, please. So the previous slide just showed, and that's okay, in 2001, that was really when the whole genome sequence was first published of a human being. There was twin publications, one in Nature and one in Science. And if you're familiar with the scientific literature, those are sort of the two of the most cited journals in science. And so one was the public consortium. So there were sort of parallel efforts towards the end where Craig Venter and a company decided to use a slightly different technology in doing the sequencing. And Celera was founded to do that. And Nature was the publication that represented the efforts that was done and public efforts that were done. Next slide. And so one thing to recognize about the Human Genome Project as it was published in 2001, it didn't represent the average sequence of a human being or even the sequence of a single human being. Literally, it was several people that donated DNA samples. Different labs worked to sequence a chunk that they were sort of given responsibility for, and then they stitched it all together. So great from an architectural perspective, it gives you some sense of how many genes that we have, but it doesn't really tell you very much about sort of what the actual sequence variability would be between individuals. But one of the things that it did one of the interesting things that came out of that was I can still remember as an undergrad prior to the genome sequence being published, we were told, oh, we've got 100,000 protein encoding genes, big genome, we're very complicated. And it turns out that we're maybe a little bit simpler than we thought we were. The complete human genome consists of 3 billion base pairs. Those are the AACs, Gs, and Ts that sort of go along, which encode for 23,000 protein encoding genes. So much less than the 100,000 that we were predicting. And so if you can, that's a huge number. So to break that down a little bit, that would be, imagine your average novel is about 350 pages long. You'd have 7,000 in your library. That would be your complete genome. And remember, every single cell would have that library of 7,000 books. So every single cell in the genome. The other surprise was only 1% to 2% of DNA actually encodes for protein, right? So the big question was, what does everything else do? For a while, it was called junk DNA because we didn't know what it did. And it turns out that it's very important in terms of the regulation of those genes. The other interesting fact is that from one individual to another, so if we were actually in a space where we were all together, I'd say look at your neighbor and you're 99.8% similar to your neighbor, regardless of what their race or background would happen to be. So we're extraordinarily similar to one another. Next slide, please. And so fast forward to 2007, and that's a picture of Craig Venter, and he was the first individual to publish his unique individual DNA sequence. So that 2007 publication was in fact an example of his personal DNA sequence. Next slide. And so what sort of happened over time is we've been able to get much better accuracy at a much lower cost. So in the 1950s and 60s, we were looking at banding patterns of chromosomes. Literally, you can get dye that would bind to different areas of a chromosome, and then you can sort of look at the banding pattern. You can see if there was big changes in terms of the banding pattern. So sometimes you can get something called a chromosomal translocation, where one piece of one chromosome kind of gloms onto another, and then you can see that the banding pattern would change as a result. So not very high resolution at all. In the 2000s, we've been able to actually look at the DNA sequence at an individual level, so an individual base pair level, so the AAC, Gs, and Ts. Big difference from looking at banding patterns. So, and the cost has gone down. We've gone from $3 billion to do the first example of a genome sequence to now less than $1,000. Next slide, please. And so this has led to, lots of promise in terms of personalized medicine. So, we know that individuals metabolize drugs differently, but the way clinical trials are run is you end up getting sort of the average dose of a large group of people. So, perhaps what we could do, or we are doing in some cases, is to actually personalize a dose for an individual, knowing that they may be a fast or a slow metabolizer of whatever drug they happen to be taking. So again, we can tailor a drug dose to the individual. We can also figure out what kind of nutritional interventions would be more or less effective in an individual. That's in nutrition genomics, and that's the bottom. That's the apple and the heart. In terms of treatment of diseases, we can, for cancer therapy, for example, we can identify what genes are mutated in the given tumor, and then we can tailor a drug regime or chemotherapy regime for that individual cancer. And finally, at the sort of the top left, that's an example of a pedigree. And when as individuals are born and go through life, we can understand what their propensity of disease would be, what they're more likely to get, and perhaps intervene much earlier to improve the healthcare of those individuals. So that's sort of the promise of personalized medicine. Next slide. So now let's talk a little bit about privacy and confidentiality. So with all of that information, there's sort of a cost associated with that. Next slide. So here's an example of a case that I had when I was a clinical fellow, and I think it identifies one of the issues with genomic information, which is that it's shared information amongst family. By definition, we share genes with our family members. So again, this is a pedigree. We've got a colon cancer. If you look at the very top, we've got an individual, a female that died of colon cancer. They had 4 children, 2 of which died of colon and breast cancer, respectively. Then if you look at the bottom line or the bottom row of that pedigree and where the arrow is pointing, you've got an individual that has been tested for a colon cancer susceptibility gene. So they don't have a disease yet, but they have a very high propensity of getting that disease. And the question becomes, that means that the other individuals that have been circled are also at a 50% risk of harboring the mutation, which would then put them at a greatly increased risk on the order of 70 to 90% of actually getting colon cancer or breast cancer during their lifetime. But the problem is that patient that I've indicated to the arrow doesn't want to actually inform her family members that she was tested positive or they may be at risk themselves. So again, nobody has the disease on that sort of bottom line of the pedigree, but the individual that tested positive with the arrow has a very high risk, and the individuals that have been circled are all at a 50% of having that really high risk. And so the question is, does the clinician have a duty to disclose, even though the patient is saying, don't disclose the information? And the clinicians tried everything. They were supportive. They said, look, we can do the disclosure for you. We can provide all that information. And for whatever reason, the patient was really quite adamant that it was her information and she didn't want it shared with anybody. And so you end up with this kind of strange situation where it's almost like public health, right? Where, what is our duty to those other individuals that we feel may be at risk? And that really is, if you were looking at what the competing principles in that case would be, it's the autonomy of the patient to make that choice about her information versus beneficence, versus our duty to care for all of those other individuals that we know are at that increased risk. So it really becomes more of a public health problem than it is sort of a conventional medical ethics problem. Next slide. So here is Dr. Watson, and so he was one of the co-discoverers of the structure of DNA, and he was the second person to have his genome sequence made public. The interesting thing about Dr. Watson is that he didn't want his ApoE status known. So the ApoE gene is known to be, there's a variant in that gene that can cause early onset Alzheimer's or increases your risk of having early onset Alzheimer's, and he didn't want that published. So he basically said, no problem, publish my entire genome, just cut out the ApoE gene, and then I'm good. Next slide. then a clever group of scientists said, well, you know, you didn't cut off a big enough piece. So what it turned, because you inherit DNA as really big chunks, you can actually infer with pretty high confidence, not 100%, but pretty high confidence, what the ApoE gene status would be just based on those flanking markers. So I've sort of highlighted just cartoon-like those markers. And because we inherit DNA in large chunks, you can actually infer what the sequence is just based on those markers alone. Doesn't matter that the sequence in the middle has been cut out. And so, again, not that many of you would have your genomes published, and not that, and if you did, would want it cut out. But I think the point here is that just because of the complexity of DNA, it's actually very hard to make those kinds of decisions, and then to think that you can keep your information private. And if anybody should have known that, it should have been Dr. Watson, should have been Jim Watson. Next slide. Here's an example of probably something that would be a little bit more germane to most of you, which would be, turns out your genome by itself is actually identifiable. There's been lots of sort of examples of that. And so I'll use the example from the 1,000 Genomes Project. So this was a study that recruited 1,000 people that agreed to make their genome publicly available. So all that would be recorded would be their genomic sequence and their city of origin, and that would be made public. No other information about the individual. It turns out, because of the popularity of genealogy databases, so if you sent a DNA sample into Ancestry.com, 23andMe does it, the National Geographic has a program that does this, and individuals are really interested in finding other people that are related to them. So you'll put, it won't be your complete DNA sequence, it'll be a chunk of it, and your last name and some contact information. That way, other people that are a close enough match can then reach out and talk to family members that they may not know exist. And so what a group of scientists did is they just compared the 1,000 Genomes database with these genealogy databases. Now they've got a city of origin. The genome is sort of the link, and then they've got a last name for very close hits. And then they were able to identify 50 individuals and actually get their phone number just based on publicly available information. So that was sort of the first example of, in theory, we thought this might be possible, but it turns out that your genome is in fact quite identifiable. Next slide. And so this has been used, the police have used this for genetic genealogy. So this is the Golden State Killer. And so, you know, this is a guy that terrorized communities for years, and they couldn't catch him. And what they ended up doing is taking a DNA sample from one of the victims, so this is from decades ago, sent it into the Ancestry.com or an equivalent, and actually got a match to a fourth cousin. And then genealogists then went in and said, okay, can we triangulate and try and figure out, based on that relative, who might have actually perpetrated the crime? They were able to find a match. So they thought they identified Joseph D'Angelo. And then the police launched an investigation. He, I think, either had a cup of coffee or ate a piece of pizza, threw out the crust. They were able to take the crust, send it to the lab, and they got a positive match. And they were actually able to arrest him and get a proper DNA sample from him. So again, an example of they were able to find him through his relatives' sharing of their DNA. Next slide. And as it turns out, if you're Caucasian, there's a 60% chance that a familial search will identify you, just because so many people of Caucasian descent have shared their information on databases like genealogy.com. Next slide. And so this has also led to certain projects. So one that I've been involved in is called the Personal Genome Project. And it actually offers no promise of confidentiality. We actually try and identify people that will be interested in sharing their data publicly. And we've got a very rigorous consent process. We actually even ask them to take an exam. We provide them a genome report. We give them genetic counseling if they're interested. And then once they've sort of made, once we've given them all the information that we have about them, then we ask them to make that choice about making their data public or withdrawing from the project. And if they make their data public, and so far everybody has, it goes on a public access database and anybody can have access to it. So the premise here, and this was really built off of a project that started in Harvard from George Church's group, was that since we can't promise confidentiality, why don't we sort of flip it around and try and identify individuals where that's a little less important to them, and then make their genome publicly available. Next slide, please. And so that's that was my little module on privacy and confidentiality. Now I'm going to talk a little bit about what data ought to be returned to research participants or patients, in terms of, now that we are able to sequence genomes and get all of this information about somebody, what ought we return? So if we've got a patient that comes in and we're trying to determine whether they've got some sort of a disorder, we can find those primary findings, right? Those would be genes that are related to the disorder that they came in to the clinic to be tested for. But what about all those secondary findings, like the breast cancer susceptibility genes, all the genes that we know have implications for health? Ought we be looking for those and returning those to individuals? Next slide, please. So here's just an example of, so each kind of pixel or each dot in that entire pie chart would represent a gene. And so, or some kind of a gene that we know is involved in something. And so there's primary gene findings. So those were genes that we know are associated with specific diseases. Those are the ones in sort of purple. So that doesn't represent a big piece of the pie, but certainly a bit of a chunk of it. Then there's genes involved in drug kinetics. So earlier on, I talked about there are genes that regulate the metabolism of drugs, and depending on whether you're a fast or slow metabolizer, you may want to adjust the dose of your medications. Then there's other genes that are sort of known to affect health, but we're not quite as confident as the primary gene findings. Then there's the genes without known health effects. And we suspect over time, as we gain more knowledge, there'll be probably more yellow and more of the colors and less of the gray, but this is sort of the state of affairs right now. And the big question is, I don't think it's controversial to say that if somebody comes in for a genetic test and we find a result and they've been counseled appropriately, we ought to give back that result. The question is, what about all the other areas of the pie, right? Next slide. And current clinical guidance is actually quite conflicting. So we've got the American College of Medical Genetics, and they have actually come out with a very strong position in 2013, where they were basically saying that anybody that goes in for clinical genome sequencing ought to get, so it's, again, we're looking at every single letter of your genome. Whether they came in for a specific purpose or not, we should actually be looking for 56 genes that are involved in cancer and neurodegenerative diseases. They're all medically actionable, so something can be done to help mitigate the problem, but that those genes should actually be screened as part of the test for whatever disorder that they came in with. So those are the secondary findings that would then be returned. Very controversially, their initial statement was that that should be done without consent. Like that, this is part of the agreement to signing up for a whole genome test, is that you should get screened for those 56 genes. They've now changed their position and said, we will accept people that only want genetic testing for the disorder that they came in for testing for, and we will not screen for those other ones. Interestingly, at the same time, and the ACMG said this should be children, adults, it doesn't matter. Everyone should have access to those same secondary findings, those 56 genes that may not be involved in the whatever disorder that brought the patient in. The American Academy of Pediatrics reaffirmed their longstanding position that predictive genetic testing for adult onset disorders should be discouraged, right? So the issue was there's a real contradiction between those two statements. So if I brought my 12-year-old daughter in for breast cancer susceptibility testing, the pediatrician ought to say, your daughter can make that choice when she's old enough to understand what the consequences of that are, because there's nothing medically actionable, during her teenage or during her younger years, right? Really, all those cancer predisposition genes really become important when you're a little bit older, when she could make that choice for herself, which is different than if she went for whole genome sequencing. So according to the ACMG guidelines, if she went in for whole genome sequencing, they ought to be testing for those mutations and returning that to parents. The Europeans took a very different approach. They just said, look, avoid the whole problem by just not screening for them. You can make choices about what you look for, and you should only be looking for the genes involved in the primary disorder that brought the patient in. That's what you should be doing from a European perspective. So very opposite to what the Americans suggested. Next slide. And in Canada, we're in between. So there's this sort of weird statement in the Canadian guidelines that say, we do not endorse the intentional clinical analysis of disease-associated genes other than those linked to the primary indication. So that's a very European sort of approach. But then they say, but in pediatric cases where labs want to report incidental findings or secondary findings, then they require sort of parental consent. There should be some notion that it could harm family members as a rationale for doing it, and there'd be no obligation to recontact. So again, a little bit of inconsistency there as well. Seems like they are discouraging looking for secondary findings, yet provide a process in place if the labs actually wanted to do that. Next slide. And so the real challenge is, in my mind, in the pediatric context. So adults with capacity to be making these choices, I think they can, with genetic counseling, can decide what kind of information ought to be returned to them. The real question is what to do with children that have emerging autonomy, and that information about their adult cancer risk susceptibility won't really affect their healthcare in their childhood. So I think there really are questions about whether that information ought to be returned to parents. And it really centers around, is returning results in the child's best interest? And so some have argued that providing that information to parents might improve the parent's health. So in other words, if you found a breast cancer susceptibility gene in the child, then chances are one of the biological parents has it as well. And if they don't know about that, learning about that through their child's genome could actually improve their own healthcare. And wouldn't that be helpful for the child? So it's much more of a relational approach. It's looking at the family as a unit versus sort of that individual child, and will it affect their healthcare? But some have sort of made that claim, and others are very much it's just about the child's health, and learning information about a parent is just sort of too far removed to impact the child's health. Next slide, please. And so here are kind of some of the ethical principles on the disclose versus not disclose in that pediatric context for adult onset variants. Best interest, interestingly enough, is used in both. So the disclosure piece, again, it's about keeping the family healthy, which then could keep the child more healthy. And that the counter argument to that is, that might change the way the parents actually think about the child, and learning about their child's genome is so far removed from any healthcare of the parents that it's very hard to draw a straight line between those two things. In terms of the benefit, these are all medically actionable variants. So, perhaps, even though they learn it as a child, this would be a benefit to them later. And then parental autonomy, where parents make decisions about their children all the time. How is this any different? On the don't release side, it's about what about the child's emerging autonomy and their ability to make those choices. And you basically remove their ability to make that choice by just disclosing to the parent. Next slide, please. So it's still a little bit up in the air in terms of what the right answer from an ethics perspective would be in that case. Next slide. And so genetic discrimination is also an extremely important concept. And it's one of the major reasons why individuals avoid genetic testing to begin with. So if you look at individuals that have, for example, Huntington's disease in their families, that's a devastating neurological disorder. There is no treatment for it. It's a dominant disorder, which means you only need one copy of the gene. And if you have that gene, it's certain that you will get the disorder. When is always the question. And a lot of people actually avoid getting tested because they're worried about genetic discrimination. And the definition of genetic discrimination, it's again usually based on the perception of the individual that's been discriminated against. And it's really differential treatment of asymptomatic individuals based on genetic characteristics. And so for one example, insurance discrimination would be sort of an obvious one, where you're perfectly healthy at the time, but you go and you want to get life insurance, and they ask you a series of questions about your health, and they say, do you have any neurological problems in your family? And you say yes, and then they will treat you differently as a result of that answer. So that would be an example of genetic discrimination. And so this is sort of a meta-analysis looking at lots of different papers on the subject. in 3 different jurisdictions, so United States, Canada, and Australia. It's an old paper, but I think it's still very relevant. One of the interesting things for me is that family and social examples of genetic discrimination were the second highest, or the highest in the United States, but in Australia and Canada were the second highest incidence of people reporting being discriminated against. So I think insurance discrimination, and I'll get into that in the next slide when we talk about Bill S201. But I think most people would sort of imagine that that could be the case, kind of the scenario that I just described, right? You have to disclose on a form that you're at a higher risk of having an illness, and then an insurance company saying, well, why would we, insure you then, or we're going to charge you crazy premiums, right? So that would be an example of insurance discrimination. But what about in the family context, where again you've got a family with that has Huntington's disease in it. You've got a child that grows up, and you've got family members that might tell that child is now an adult. You probably don't want to have a family or get married because you could pass along this mutation, and wouldn't it be terrible to have a child and then you have to subject them to having all these neurological symptoms and dying prematurely. And so that would be an example of genetic discrimination in a family context. So again, very surprising to me that was I was expecting insurance discrimination, maybe employment discrimination, but I wasn't expecting discrimination from one's own family members as being such a high incidence. Next slide. And so we've actually got a fairly good bill in Canada. So Bill S201. It's called S because it was actually written in the Senate, and they don't write many bills. That's kind of an interesting side note to all of this. But basically, it prohibits any person from requiring an individual to undergo a genetic test or disclose a genetic test result as a condition of providing goods or services, contract. So certainly insurance would fall under sort of the contract, right? So an insurance company can't actually ask or give differential treatment based on you offering up a genetic test result. They also made amendments to the Labor Code and the Human Rights Code sort of to codify the fact that you can't discriminate based on somebody's genetic background. And so this was passed by the Senate in 2016. Next slide. There's actually a Supreme Court challenge, which was recently overturned. So Quebec had argued that this infringes on provincial jurisdiction. So things like insurance are actually regulated at the provincial level, and the government of Quebec was arguing that this was an infringement on a provincial jurisdiction, and the Supreme Court disagreed. They upheld Bill S201. Which made a lot of people happy, except for the insurance industry that really felt like this interferes with their ability to make money. What insurance companies are worried about is a term called adverse selection. So they're worried that somebody will know something about themselves, about maybe having a high risk of a certain cancer. The insurance company doesn't know that risk. So they can't adjust your premium accordingly. And so you may load up on insurance knowing this information. The insurance company doesn't, it's not an equal playing field because the insurance company doesn't know, and you end up costing kind of more money than they ended up allocating. And so that's sort of the insurance company's concern. Although, Mendelian disorders are so rare in the population, there was actually an analysis done that really isn't a valid concern. And the other aspect to this is that, so yes, they can't discriminate you based on your genetics, but they certainly can based on your family history, which is kind of the same thing. And so another criticism is that Bill S201 doesn't actually go far enough, since insurance companies can still discriminate based on your family history. So they may not know what your genetic predisposition is, but if they know that you've got a first-degree relative that died of early onset breast cancer or colon cancer, then they can pretty easily surmise that you've got some kind of a mutation likely, or your risk of having a mutation that could put you at increased risk. And so then they can discriminate based on that information alone. So it's still not perfect. Next slide. And so maybe just to finish off that piece, other jurisdictions have done things a little bit differently. So for example, in the UK, everyone can have access to, I think, £500,000 of life insurance. maybe that's a little bit high, but there's between 100,000 and 500 pounds of life insurance without having a medical. And that has been argued as one way of also leveling the playing field in terms of not discriminating against people based on their background. So now I'm going to switch gears a little bit and talk about some recent developments in our ability to actually modify genomes and what that might look like. Next slide. And so 2020, these two remarkable women won the Nobel Prize for chemistry for something called Crispr Cas9 the genetic scissors. So they were able to figure out how to actually target specific areas of the genome, which was something that wasn't done before. And so Emmanuel Charpentier and Jennifer Doudna are sort of co-discoverers of this process. Next slide. And so basically, if I'm going to talk a little bit about the science in the next couple of slides, and I recognize that some people get it faster than others. So if all you remember is that Crispr Cas9, and CRISPR stands for clustered, regularly interspaced, short palindromic repeats. So if all you remember is Crispr Cas9 is like genetic scissors and it can make specific cuts in a human genome, that's sort of good enough. I'll get into a little bit more just because it's really interesting, but whenever I say CRISPR Cas9, you just think genetic scissors, that's kind of good enough for the ethics discussion that's going to follow a bit of the science. Next slide, please. So it turns out bacteria have an immune system, and CRISPR Cas9 is part of that immune system, and we've now exploited it into and using it in the mammalian system. And so just like in our immune system, one component of our immune system are antibodies, right? So if you get sick, you'll create antibodies to whatever virus invaded your body, and it will attach itself to specific proteins made by that virus, and your body will recognize that as something that needs to be dealt with, and again, those particles will be destroyed. So the bacterial immune system works very similarly, except instead of having antibodies, it uses DNA. But again, the DNA sequence is kind of analogous to how we use antibodies in our system. So every time a virus infects a bacteria, it actually spits in some viral DNA. And so that's a picture of a virus in the sort of the top right there. And it's injecting its viral DNA into the bacteria. And if the bacteria survives the infection, it'll actually keep that little piece of DNA, and it actually is put into the CRISPR sequence of the genome, of the bacterial genome. So that's actually added to every time it gets infected by a different virus. It adds a little piece of that viral DNA to the CRISPR region of the DNA, and that CRISPR DNA then makes RNA. Next slide. And again, that's just by the normal machinery of the cell. So this is just the cell, the bacterial cell that's actually making the CRISPR DNA, and then it makes the RNA from the DNA. And then this next slide is a little bit complicated, but all you really need to know is that it actually breaks off little chunks of those viral DNA particles, which can then bind to new particles that might be infected into the cell. So when you've got a new infection, or you've got a different virus that's now infecting the cell, if it's the same sequence as a previous virus that infected the cell, it'll get bound by those genetic scissors, and it'll basically cut up the viral DNA sequence of the DNA that was just injected into the bacteria. So in essence, the bacteria has a memory of all other viral DNAs that ever infected the cell. And if it recognizes any of the viral DNA as being similar to that, it can then bind to it, and then there's enzymes that just chews up the DNA. And if the viral DNA is chewed up, then it can't actually make proteins, and it can't actually infect the cell. So it effectively acts like an immune system. Next slide. And so it turns out, again, based on the work of those two remarkable scientists, we were able to figure out a way of exploiting this in the mammalian genome, where we could now create a guide RNA that will bind to a specific region of the genome and make a specific cut, right? So we've been able to make cuts in the genome before, but not in specific ways like this. And so basically what we can do is either remove a piece of DNA, and that would inactivate the gene, or we can then remove a piece of DNA that perhaps has a mutation in it, and then repair it. We can stick in a new piece of DNA that has the normal sequence in it, and then effectively repair a genetic mutation. Next slide. And so there's other ways that we can do this. So this is one example. So the CRISPR Cas9 is kind of the new sexy way of doing this. But there are other modalities called gene therapy would be one. And these are viral vectors. And so there's a picture of those viral vectors. And this would be a case of a blindness caused by retinitis pigmentosa. And so that in these cases, there's specific gene mutations that lead to retinitis pigmentosa. And there are ways that we can actually repair the genome. So we can fix the mutation such that the normal proteins are being made again, and then those cells can recover. So there's several different types of diseases that are now being treated using gene therapy, but arguably CRISPR Cas9 offers a much more sort of effective way of doing this. Same kind of principle. Next slide, please. And so the critical determination here is the somatic versus germline. So your somatic cells are every single cell in your body except your gametes, right? So sperm in men, eggs in women. And so really the critical ethical question is, if you're changing the germline cells, then you're actually not only changing the cells of that individual, but you're changing the DNA of all subsequent generations. You're making a permanent change in the DNA of anybody that's sort of downstream of that individual. Changes in somatic cells, and so the example of the eye that was getting an injection previously, that would be an example of a somatic cell, right? So we've got a cell that's already been born in an adult or in a child. It's not working properly, and we're trying to fix it. So really, CRISPR or gene therapy in somatic cells are very similar to how we would administer a new drug, right? So there's clinical trials, there's an entire apparatus in terms of how to deal with that, in terms of how do we safely use genetic technology to combat disease. What's very controversial is how do we do that in germline cells? So one argument could be made, well, if we know what the mutation is, and there's a way to fix the mutation, then rather than have this problem keep reoccurring every generation, why don't we just fix it and move on, right? The counter argument to that, of course, is that there may be all kinds of other off-target effects that we may not know about. There's safety. There's ought we be messing around with the germline that way? We evolved in a certain way, and ought we be messing around with nature? That's kind of the rough sketch of the counter arguments. And so next slides, please. So I'm going to focus on the germline situation because I think that's the more controversial one. That's the sort of the harder nut to crack, so to speak. I really think about the somatic cells as being just very similar to how we would, again, test a new drug. And so this really kind of broke in November 2017. You may have read this in the popular press, or the scientists amongst you in sort of the scientific academic press. But this is a scientist trained in the US, has his lab in China. And so Dr. He revealed that he used CRISPR Cas9 on embryos and then later implanted those embryos and actually had 2 babies born. Next slide. And this is a slide presentation from his presentation. And so essentially what he did was he inactivated, and there used to be some animation here which didn't work, but so where you see sort of the big X crossed out, so that's HIV, and for HIV to enter a cell, it binds the CD4 and the CCR5 co-receptor. And that will then, once it binds to those receptors, it's then brought into the cell and then it infects the cell. And so we know in the population, there's some individuals that have a mutation in the CCR5, naturally occurring, that actually makes them immune to HIV. And so there's sort of fascinating data on, there were some individuals that were very high risk for HIV transmission. So IV drug users, sex workers that never got infected with HIV, even though they were almost surely exposed. And they were able to figure out that in fact, it's this naturally occurring mutation in the CCR5 gene that made them essentially immune to HIV. And so of course, changing one thing causes other effects. So If you don't have the CCR5 gene, you're more likely to be at risk for West Nile disease, and there's several other disorders that you can get sicker. So it's always going to be a trade-off. But essentially, what Dr. He did is he had a couple. One was the male had HIV, the woman did not. And he essentially, using in vitro techniques, harvested sperm, harvested eggs, created embryos, exposed them to Crispr Cas9 that was specifically to remove a sequence in the CCR5 gene, and then took those embryos and transplanted them back into the mum, and then she gave birth to Lulu and Nana, which aren't their real names. Next slide.
Michael, this
Yes.
is your 4-minute warning.
Is it really? Okay. Okay. I know what I'm going to do. Okay. So his experiments were widely condemned. He didn't address any unmet medical need. There are ways of avoiding HIV without needing to inactivate any genes. The gene editing wasn't in fact executed very well. There was mosaicism, which means that there was different cells in the embryo. Some had the mutation by the Crispr Cas9 and some didn't. So it wasn't like the entire embryo, in fact, had the same genetic makeup. They were now different genetically from one another. And there were examples of off-target mutations. So the Crispr Cas9, although specific, sometimes makes mistakes. And there were examples of at least 1 or 2 mistakes. It's not clear what those new mutations will do. There were certainly big problems with the informed consent process because the parents didn't really understand what was being done. They thought they were enrolling in an HIV trial, not in something that would change the genetic makeup of their babies. And he certainly contravened a global consensus against doing this. And he sidestepped all research subject requirements. So there was no REB approval. There was, again, not an informed consent process. He didn't do all the necessary preclinical work to show some safety. And so he really, it's hard to emphasize, if you can think of an experiment that would do everything wrong, this would be it. Next slide. And so there's lots of guidance documents, especially as a result of this that came out. I'm going to maybe just focus on one to end, the Nuffield Council on Bioethics. Next slide. And really what they focused on in quite a detailed report was sort of 2 ethical principles. So one is the welfare of the future person. The one interesting aspect of Crispr Cas9 in a germline therapy context is that it's done on somebody that doesn't have the ability to consent because they haven't been born yet. And so there's a real question about the therapy versus the enhancement. So we can treat a disorder, or we can perhaps enhance the individual and make them smarter or taller, or you can pick whatever a genetic trait you may want to improve. There's certainly there's a diversity and disability critiques about that, equity and justice in terms of who will get access to that. Social justice and solidarity in terms of this may solve an individual family's problems, but is it really good collectively for society, right? If we become, if we start making decisions about, what we ought to look like. And again, focus on social harms as sort of, again, a precondition to being able to do this properly. Next slide, please. And so the report basically said, there shouldn't be categorical limits. So we shouldn't ban the use of this, but we need to have conditions. It can't be biologically reckless. They need to be consistent with the welfare of future people. It can't be socially divisive, and it shouldn't be initiated without prior societal debate. Next slide, as a precondition to doing this. I'm going to actually skip this section because it's very similar to the first. And so really the focus has been on the application, not the technology. So there have been groups that have actually suggested that we've got a moral imperative to actually do this. Whether it's for enhancement, single gene disorders, future generations, we actually have a moral imperative, if this is going to make people better and make society better, to actually use this technology. And again, it's all about the applications. Next slide. There's been some religious objections. So certainly about the disproportionate risk, the potential loss of embryos, which again are considered human life with full moral status in some religious circles. The use of reproductive techniques, which again, so for example, the Catholic Church does not promote the use of any reproductive techniques. And again, the enhancement that would be immoral. So the use of CRISPR Cas9 to treat disorder would be, again, consistent, but using it for enhancement would not. So again, there are religious objections from various groups as well. Next slide. There's also legal objections. So for example, Canada actually has a ban on using CRISPR Cas9 for changing germline DNA. And this is just a slide representing different countries in the world. And there's different, so some are prohibitive. So for example, Canada and Brazil, some sort of intermediate where there's policy documents, and there's a lot of countries that actually don't have any legislation at all. Next slide. This is one of my last ones. So doing this sort of technology would violate the Human Reproduction Act. And this is the prohibited activity is you cannot alter the genome of a cell of a human being or in vitro embryo such that the alteration is capable of being transmitted to descendants. Another fancy way of saying you just can't do germline editing, full stop. Next slide. So in summary, and I think I've actually managed to finish on time, whole genome sequencing will soon be widely available to both clinicians and consumers. Genomic information in and of itself is identifying. I don't see that as being a problem, but people need to be aware of that. There is disagreement about secondary findings and what info should be returned. Genetic discrimination is the differential treatment of asymptomatic individuals. We do have legislation in place in Canada, although it doesn't stop insurance companies from discriminating based on your family history, and it certainly doesn't prevent family members from discriminating against individuals. And remember, that was one of the highest incidences of genetic discrimination. So the law will not fix that. And genome modification, it's coming, and there really have been calls to, like, how do we because this, one of the things about CRISPR Cas9, it's actually quite cheap. So any lab can do it. And so how do we ensure that we've got some kind of a regulatory scheme that protects the human species and protects them from people that might want to try things out in ways that they shouldn't. And I will stop there. Thank you so much for your attention and time.